Hello guys, my name is Dr. Varun and here I am back with another day, another 30 most important topic for NEET 2025 biology.
Chalo, let's complete it.
First of all, sorry for yesterday that there was no video, and for the past 3 days there was video at very odd time because I was little stuck with the network issues. But from now on, continuous videos will be there. Plus, as we said that every day one video will be there, so the video which is not being completed will be uploaded tomorrow as an extra video. Meanwhile, let's just come up on the today's topic, that is we gonna be studying about the sliding filament theory and a very important question that will come from it, which we will try to decode. Moving on to this theory. Now just think that if your muscles wants to contract, of course I don't have any muscles, so you can call me Mr. Skinny Sir, but if you are contracting your muscles, means contracting of muscles means you are doing any movement which is related to muscles. Now how it is taking place. Now basic question, like if I want to do this with my finger, then means what my brain will think about it, from where the message has to reach this part. From brain it will reach to spinal cord it will reach to the hand spinal nerve, and finally it will reach to this part, and then it will say that okay, do like this, correct? So this whole thing is what your brain operating it, means it has somewhat neurons with it. So everything starts from that. There is a one nerve impulse that will reach here. Since the nerve impulse is doing a motor function, motor means moving, motor. Okay, so motor cycle is what? Moving. So you have to move these hands and all, move these hands and all, means motor function is there, the motor neurons will be working. So a motor neuron will bring the stimulus of what? That to move like this, the muscle contraction at this end. Okay, so starting with there is a neuron, so we will make a neuron first. So imagine that this is the neuron, the end neuron. Of course it is a nerve fiber, which big one, but at the end what is happening? It is at the nerve neuron. So this is, we will call it what? Motor neuron. Why motor neuron? Because since it is doing what? Motor function. So this motor neuron, it brings message from brain. Now over here what? There is a presence of muscle fiber. Okay, so the muscle fiber, if you remember, it is what? A cell. So we used to make it what? Long cylindrical structure. Okay, so this is what? Our muscle fiber. Muscle fiber is nothing but muscle cell. So muscle cell is called as what? Muscle fiber. Okay, so this is your muscle fiber. Now in this muscle fiber, now if it is a cell means what? All the organelles could be present. So can I make that there is what? There is nucleus present. I have told you that it is what? Multinucleated condition will be appear in it. So I am just making you look like this, that there is a nucleus present. After nucleus, with the nucleus what is present? There is a presence of endoplasmic reticulum. Of course other things are also present. I am making endoplasmic reticulum. Now here what special thing about why I am calling endoplasmic reticulum? Because it has a name with it. Okay, so what is the name of this endoplasmic reticulum? The name of this endoplasmic reticulum here called as sarcoplasmic reticulum. It is important to remember. Plus, since it is a muscle cell, so the muscle cell plasma membrane has also a different name called as sarcolemma. Cytoplasm of this will be called sarcoplasm. Okay, just remember this thing. Coming back, a neuron is coming, a motor neuron is bringing a message from the brain. Now what will happen? If you remember from the little bit about the depolarization, repolarization, so terms will come. If you're not able to understand, we will do that in one of the video, because that is also important one. Then you can come back and see this video again, but here just try to understand the basic. So over here inside it what? There is a presence of synaptic, like in the last node, that is synaptic vesicle. So these are what? There is presence of synaptic vesicle inside these vesicle there is the presence of neurotransmitter. So this neurotransmitter will be what? It will be released in this area. Now this area where this neuron is present, this muscle fiber is present, we don't call it synapse. We call it what? Neuromuscular junction, because it is an area where neuron is also present, muscle is also present, and it's a junction where they are meeting. Okay, so neuromuscular junction. Okay, next part, it is also called as motor end plate. Okay, means this is what? Motor neuron present over here, and this is the end area of motor neuron. So we call it motor end plate. So what will happen? This, when the impulse will come, the second step would be what? Second step would be neurotransmitter is released into synaptic cleft, and this area is called as what? It is called as neurotransmitter junction, or it is called as motor end plate. Now there is a one thing we have to remember. If the neurotransmitter is released, for example, if it is acetylcholine is released, so what will happen? If just see that it is acetylcholine, so what will happen? This acetylcholine has to be, first it will be degraded into the acetate and choline enzyme. Okay, and then with the help of an enzyme, and then it will go and attached to the plasma membrane. So means third step would be what? The neurotransmitter, whether it is excitatory or inhibitory, since this one acetylcholine is excitatory, which will do the function. Okay, won't stop the function. So the neurotransmitter will act as stimulus and it generate action potential. Okay, this thing is there. Now if you see something extra I am telling you, which if you don't see, that is also okay, but if you see it, very good, you will become beyond the NEET students. So what is that? It is that this plasma membrane, you know what, it is little bit invaginated. Why? So that it can go deep inside the cell. Okay, so that it can work on sarcoplasmic reticulum. Okay, just remember that. And this thing is also called as T-tubules. Okay, extra, not need to remember. If you don't remember, that's okay, fine. Okay, at NEET level, for UG level, you need not. Okay, so the action potential that will generate, if you remember a little bit, what it does, it will what? It will change the polarity. And what will happen? So the next step would be what? Next would be depolarization. So when the depolarization, what will happen? Rapid influx of sodium will occur. Sodium influx. And we call it membrane is what? Depolarized. Okay, moving further, this sodium will do what? This sodium will go and act on sarcoplasmic reticulum. And here we understand that why I was emphasizing on sarcoplasmic reticulum. So this sarcoplasm, it act on this. And as it act on this one, what will happen? This sarcoplasmic reticulum, it start releasing the calcium ions. So can we say that in the muscle, sarcoplasmic reticulum is the storehouse of the calcium. So calcium will be released. Now let's see. In this muscle fiber, there are contractile proteins present, which is what? An important feature of it. That muscle fiber have contractile proteins. Spelling, let it be, okay? We will come back. Now please, anyone don't commit for it like what is this spelling is wrong and all. I am writing very fast. Okay, so contractile proteins are present, which are actin and myosin. So their arrangement is what? In the form of sarcomere. Okay, so these contractile protein is present in the form of sarcomere. So sarcomere is alternative proteins, two different types of proteins in the thick and thin filament segments they are present. Means there are two types of protein, myosin and actin, along with tropomyosin and the troponin. So they form what? They form bundle up together, and one appears thicker in appearance, other appears thinner in appearance. So let's just see that if something is thick, it will appear darker in color. So I am making here a darker kind of a protein. So this red one protein, contractile protein that is present everywhere. I am making it here, spaceless now. So it is present like this. So this is what your darker protein. Simultaneously there is present what? Lighter protein also. So lighter protein I am making it like this. Okay, like this. This is 3D structure. I am just making a 2D structure. Okay. Multiple layers are present, their arrangements are there, which from here a very important question will come. Okay, so you should know that how many actin protein or thin filament or thick filament is surrounding one of vice versa. Okay, if you don't understand this question, I will write that in comment. Just write it that we don't understand. Okay, that's totally okay. Coming back to means these are the segments present. Now in this, how much is one sarcomere? In one sarcomere, what is present? That this half of the thin filament and full of the thick filament. This is representing one sarcomere. Okay, I assume that you know sarcomere, but here I am explaining it again. So what happened if you see the sarcomere, how a sarcomere will look like? So we have thick filaments like this, and we have what? Thin filaments like this. Okay, this looks thick. This is what? Thin filaments. Now understand what are the terminologies, then we will able to do one of the very important question which has higher chances of coming, conceptual question. Okay, now the thick filament, this area I am marking, okay, from here to here is called as A band. Okay, sarcomere's A band. Now the A band has only center part, if you see, that is what? Made up of only the thick filament, that is myosin, whereas this area of the thick filament is made up of what? This structure also and this kind of protein also, means both the proteins are present. So the area where only single protein is present, that is myosin, okay, is present, this area is called as H zone, and from this H zone a imaginary line going, which is actually what? Protein line going, that is known as what? This imaginary line we call it M line. M for middle, no? So M line. Okay, now simultaneously we were studying that what is this? There are certain part of A band which is being overlapped by the thin filament. Okay, so this area is that, this yellow color one, this area is called as what? O zone. O mean overlap. I will remember it like that. So O zone is there. Then someone will ask, right, what is about the thin filament which is present inside it? The thin filament that is present over here is known as this whole structure as I band. I band. And in the mid of the I band, there goes what? There goes a imaginary proteins line which is dividing the I band into half. This is called as Z line. So if someone will ask, like, what is a sarcomere? It is a distance between two Z lines, okay, that represent one sarcomere. Okay, so half of the I band, half of this I band, and full of the A band makes what? A sarcomere. This can also be called. Now let's just come back what happened over here. So these bands are present. Now what happened? This band has what? A head-like structure present like this. If you remember the proteins. And on this thin filament, so I am going on the next page. Okay, so what will happen? That there is what? If you remember, the thick protein has what? A head-like structure. On this head-like structure, two sites are present. What are the sites? One site is the ATP binding site. With this, what happened? A ATP molecule is binded. Okay, so ATP molecule, I'm making like this. A ATP molecule is always binded to it. Okay, at the resting stage. Whereas another point, this, it is what? It is the actin binding site. This is actin binding site. Means this site will make with the actin. Whereas your thin filament, so this is what? Multiple of them are present together and forms what? They forms the thick filament, which is red in color I have made. Okay, thick filament. Whereas the thin filament, how they look like? The thin filament have multiple structures. So first of all, it has what? Actin filaments. So if the actin filaments are present, means it is made up of actin. So if you see this one, this is your how a actin fiber look like. So basically they are globular proteins, means circular in appearance, but they stick together and form what? A bead-like structure. So this is what you are forming F-actin. Individually they are called as what? G-actin. So G-actin polymerize. Polymerize means monomer unit join together and form F-actin. Whereas there is what? There is another proteins. Not just this is the in the thin protein present. There is a presence of tropomyosin. This tropomyosin is also double and it is also helically present around there. On the top of this, there is a presence of troponin, which are a 3 protein consisting structure. So this is what? This is your troponin. And these forms all what? They all are forms what? Thin filament. So thin filament has 3 different types of protein, whereas thick filament has only one myosin type of protein. One point we have to remember. Coming back to see the sarcomere and how it is working and how these terminology is working. So myosin has what? Myosin is what? Thick filament, and your actin, troponin, and tropomyosin is forming what? Your thin filament. Till here, okay. Let's just see that how the contraction occurring, means again we going back to what? This thing. So I am making it over here. So if you see, there is what? Thick filament present. So I am making thick filament like this. Small one. Okay, this is thick filament. And then we have the what? Your thin filament beta. So thin filament is present like this. Able to see? Now this thick filament has what? Head-like structure like this present. Multiple of them are present. So till now what I said? I said that calcium is released. Now here we will learn with best of my way. What is that? It is a love story. So in a love story, what is needed? A boy is needed, a girl is needed. So imagine that over here, this is what? Our thick filament. This thick filament is called as what? Myosin. Our myosin is what? Man, boy. M for myosin, man, boy. Okay. And simultaneously, this actin is what? Our female. So this actin is our female. Imagine it. Now, in a love story, okay, you need what? You need some masala and drama. So who create masala and drama? Of course, family. Okay, so here the myosin mama is ATP, which do not want that myosin go and meet with actin, because until they will meet, there won't be any contraction occur. Means they won't come closer. If they won't come closer, then what? This not happen, this not happen, this not happen. We want that to happen. So now what will happen? Similarly, do you think that my actin parents want to do it? They also don't want it. Who is their parents? That is troponin. Tropomyosin is also working, but troponin we have to remember. So actin parents is troponin, whereas, so I call them brothers because brothers, females, brothers don't allow. Okay, so actin is a female. This has what? Troponin. 3 troponin are what? The brother of the actin. So they don't allow the female to meet the actin. Now both actin and myosin want to meet. So what will happen? Here comes the role of friends. So be a good friend. Okay, don't show this to your parents. They will definitely put a strike on my channel, but that's how I let you know that. So you need a what? You need friends. So who is the friend? Calcium. Okay, so the calcium which was released by that remember that transmitter and all those things which we studied, that work. Those calcium goes and bind with what? With the troponin. And just remember what is happening with the troponin. So just imagine that if someone for example, I am here and my brother is here. Imagine actin. So if someone will come and say that, brother, can you tell me, like someone came, calcium came that, tell me that what is the address to this area and all. So troponin is just little bit, keep their eyes away from me and this actin found it correct time. Meanwhile, the same time, the mama of the myosin, that is ATP, has to be released. How it will released? It will released by the help of what? By the help of ATPase enzyme. So the friend of myosin is what? ATPase enzyme. It will go and it will break the ATP. Means hydrolyze the ATP. The ATP will change into ADP and P. And as it change into that, what will happen? I told you that mama is stopping it. So mama is not present there. Mama has been broken down. Okay. So what will happen? This myosin will bind with the actin. So myosin head, imagine this is the myosin head and this is the actin. It bind with it and it will pull it closer. So as it pull it closer towards inward movement, the contraction occurs. Let's just see that in this term also. So if we are writing over here, if you remember that we have written certain steps over here till this. Now the calcium is released. So sixth step was what? Sixth step was calcium released from sarcoplasmic reticulum. Next point, that is the 7th point. Okay, so the 7th point say that the calcium released, calcium will bind with troponin and that will unmask or means unhide the site of myosin binding present on actin. So actin is free now. The girl is free, that she will definitely go. Okay. Simultaneously, ATPase enzyme hydrolyzed ATP present on ATP binding site of the myosin. Okay, now what will happen? Since this leads to unmasking of actin binding site on the myosin. Actin binding site on myosin. Okay, as they both are free, these sites are free, they will join. They will make bond. And the energy released, because when the ATP will hydrolyze, energy will released. We know this fact. So the energy released will be utilized in power stroke. So what was power stroke? I told you that if this is the actin head, sorry, this is the myosin head and this is the actin, it will bind to it and it will pull it towards. This is called as power stroke. Okay, in power stroke leads to contraction or pulling of the actin filaments towards inward. Okay, contraction leads to contraction of sarcomere. Simultaneously it is occurring everywhere, and if it will occur everywhere, what will happen? Your muscle will be contracted. Till the time calcium is present, this contraction keep on happening, like once the, what will happen? After that ATP is being hydrolyzed, after some time mama will come back, na? So another ATP come and join, it will stop the bond, okay, it will break the bond and it will come back to natural. But calcium if still coming, then mom will again be deflected, sorry, this brother's troponin will be deflected, ATPase will again deflected, this will again make the bond, again power stroke, contraction keep on happening. Until when? Until there will be an inhibitory sign comes which will make the calcium back again inside the sarcoplasmic reticulum. This is it. Now where the question will come? The question will come during the contraction, what happens with the sarcomere? Okay, so this becomes the important topic for you for today. That is what? During contractions. During contraction. So if you remember about the sarcomere, if not, then go back, see that again and make it yourself. You should know that A band, this zone which is being. Now imagine that something is being made if the contraction is occurring. Contraction will occur, what will happen? It will go closer. So if you remember, there is one diagram given in your NCRT, which I'm going to show it to you. This diagram, which is telling you at the relaxed state, these two sarcomere, how it will be, when it will be contracted, it's coming closer. So you have to use the scale or of your visual understanding to remember it. What it is, that when the contraction will occur, the size of A band remain constant, means A band will never become shorter. Okay, remains constant. Okay, then what about the size of I band? I band size becomes shorter. Okay, so I band reduce in size. Okay, then there is what? There is a H zone. Now depend if something is being pushed inward, it will come in the area of H zone. Okay, if you will see. So if you don't remember, then just see this, remember this fact based. So the size of H zone, where only A band was present, because since I will go inside, bombard inside, okay, so it of course not bombard, okay, so size of H zone will increase not increase, size of H zone is also decreases, that is reduces. Whereas overlap zone, since overlapping is increasing because I is coming closer inside, okay, so the size of O zone, it will increases, whereas size of sarcomere, it will reduces. That's it. Okay, so that is the most important topic which you have to remember. If you don't remember these pointers, you have to remember for your NEET examination 2025. Well, that's all for this video. I hope you like this video. It's little tough. I try to make it fun and interactive, but since the time frame was less, I can't. But if you didn't understand it, tell me again. I will redo this topic in more engaging way. Okay, well, that's all for this video. If you like this video, give it a like, subscribe this channel and share it with your friends, or what I say always, maybe not, because you need to clear your NEET exam. You didn't take a thing for your friends, right? So that's all for this video. I taken over. Bye.